In a quiet laboratory outside Johannesburg, a freezer holds tubes of saliva and blood drawn from more than 5,000 living South Africans. Their genomes were sequenced, sorted, and published in Nature Communications in April 2021. The study did something nobody had quite managed before. It showed, in measurable statistical terms, that the descendants of the men who broke a British column at Isandlwana on the morning of January 22, 1879, are not metaphorically alive. They are continuous. They walk the same valleys their great-great-grandfathers walked, carrying the same Y-chromosomes, the same mitochondrial lineages, the same long inheritance of land and language. There is a second thread to this story. The warriors who charged that British line were not, in any ordinary sense, sober. They had been doctored, and recent chemistry has begun to identify, molecule by molecule, exactly what was running through their blood. To understand what the British walked into on the morning of January 22, 1879, you have to go back several days and several miles east to a place called Ulundi, the royal capital of the Zulu kingdom, the seat of King Cetshwayo Kampande, who in 1879 was around 50 years old and had been on the throne for 7 years. By the time Lord Chelmsford's columns crossed into Zulu territory, runners had already reached Ulundi. The king had summoned the army. Roughly 25,000 men were gathering at the royal homestead, and what happened to them in the days before they marched is the part of the story the British never understood and never quite managed to record. The Zulu army did not go to war. It was sent to war, in a precise technical sense, by the king's doctors. They were called izinyanga. Their work took several days, and it was begun the moment the regiments arrived at the royal kraal. The sources describe it as the strengthening of the army. In practice, it was something closer to a controlled chemical and ritual transformation, performed on tens of thousands of men in sequence, designed to bring together, in the period phrase, the hearts of the people. The first stage was purification. Each warrior drank emetics and vomited, communally, into a single great pit. The body was emptied. The next stage was the bare-handed killing of a black bull, slaughtered without weapons by warriors of one of the regiments, its carcass disposed of according to prescribed rules. The bull's strength was understood to pass into the men. Then came the sprinkling. The izinyanga prepared liquid containing material particles called insila, which were said to belong physically to the chief whose people were about to be attacked. Hair, nail parings, spittle scraped from a wooden bowl. Secret messengers had been sent out beforehand to obtain these substances, and the war could not properly begin until they had been collected and infused into the doctoring liquid. Each warrior was sprinkled with this preparation, and so was each spear, each shield, each leather skirt. The intent was specific. The enemy, by ritual logic, had already been bound into the army's medicine. There were further stages. The warriors were given to drink and to chew and to inhale a sequence of plant preparations that will be described in detail in the next section. They were marked on the body with the residues of these substances. They were given doctored beer. They were chanted over. They were instructed in the dreams they had brought back from the previous nights of preparation, and the izinyanga interpreted the dreams as omens. By the time the regiments were finally released from the royal kraal and turned toward the British, several days of synchronized ritual, dietary, and pharmacological preparation had passed. The men who set out from Ulundi were not the same men who had arrived. Their nervous systems had been worked on, their stomachs had been emptied and refilled, their fear had been ritually transferred into the bodies of cattle and the substance of an absent enemy chief. They walked roughly 70 miles in 3 days, in disciplined silence, eating little. By dawn on January 22nd, they had crossed into the high country and were lying flat in a ravine called Ngwebeni, a few miles north of a sandstone hill the British called Isandlwana. They were not seen. A British scouting patrol stumbled on the edge of the formation by accident around noon. The valley moved when they did, and 25,000 men rose from the grass. Lord Chelmsford was not at Isandlwana that morning. He had divided his force the day before and ridden out with half of it to chase what he believed was the main Zulu army somewhere to the southeast. He left fewer than 2,000 men in camp. He had not constructed defensive works. He did not know that every man hidden in the Ngwebeni ravine had been pharmacologically prepared for the moment of contact. The British did not know what they were facing. We are only now beginning to. The first substance to name is Intelezi. The word does not translate cleanly into English. It is not a single drug. It is a category of ritual plant preparations gathered and combined by the izinyanga, the herbalist, according to recipes that varied from healer to healer and from purpose to purpose. Some Intelezi were sprinkled, some were drunk, some were rubbed onto weapons or shields. A 1998 ethnographic sample collected from an izinyanga in KwaZulu-Natal contained, among other ingredients, a silvery substance said to produce bright flashes in the eyes of an opponent, distracting him at the moment of engagement. Other Intelezi were said to cause dizziness, strokes, or impaired vision in the enemy. The 19th century missionary Henry Callaway recorded Zulu informants telling him that men trusted Intelezi very much, that a man traveling without it felt naked, and that a fighter who had washed in powerful Intelezi could injure another man simply by playing with him. The second substance is Dagga, the South African strain of cannabis sativa, smoked in long pipes, drunk as a tea, occasionally chewed. The active compound is delta-9 tetrahydrocannabinol, the same molecule found in cannabis everywhere, but the local cultivars carried high concentrations. The ethnographer Alfred T. Bryant, who lived among the Zulu in the late 19th century, recorded watching warriors smoke Dagga before combat and wrote that afterward they acted, in his phrase, like they could do anything. The pharmacology is dose-dependent. At low doses, THC is sedative. At high doses, in combination with adrenaline and group ritual, it produces dissociation between pain and the body's response to pain, a flattening of fear, and a willingness to move toward danger that under any other circumstance would seem inhuman. Some Zulu warriors carried Dagga with them into battle. Some smoked it in the hours before engagement. The third substance is the most striking, and the one most directly traceable in modern chemistry. It is called Buphone distica, a member of the Amaryllis family, known in English as the Bushman poison bulb. The Greek name Buphone means, literally, ox killer. The bulb contains a class of compounds called isoquinoline alkaloids, the most potent of which is bufonadrine. Bufonadrine is hallucinogenic. It is also a powerful analgesic. The botanist Ben Erik van Wyk, of what is now the University of Johannesburg, has noted that the dose of bufonadrine required to produce pain relief sits dangerously close to the lethal dose. Only experienced traditional healers handled the bulb, and they tested each preparation on themselves before administering it. The Khoisan, and older people of the same region, used the same plant to embalm their dead and as a doorway, in their cosmology, to the world of ancestors. The fourth substance was uchwala, sorghum beer, doctored with herbs by the izinyanga and given to the warriors in quantity. It was believed to render them resistant to enemy weapons. Whatever the ritual claim, the actual effect was alcohol intoxication layered onto an empty, vomit-cleansed stomach, multiplied by the absorption rate of dehydrated men. The fifth, more disputed, is Amanita muscaria, the fly agaric mushroom, whose principal psychoactive compound is muscimol. Muscimol is a GABA receptor agonist. It produces dissociation, altered perception of size and distance, and a particular kind of fearless aggression that has been documented in Siberian warrior cultures and that period observers reported in some Zulu sources as well. Whether Amanita muscaria was reliably available to the izinyanga is still debated. Now compress all of this into the body of a single warrior at Isandlwana. By the time he crested the ridge, he was likely carrying delta-9 tetrahydrocannabinol from Dagga, an isoquinoline hallucinogen with analgesic action from buffone, ethanol from doctored sorghum beer, and the residue of communal emetics in his gut. His skin had been sprinkled with Intelezi containing material from his enemy. His shield had been doctored, his spear had been doctored. This was not recreation, this was applied chemistry, refined over a span of time so long that, as the next section will show, it predated the Zulu kingdom by approximately 60,000 years. In 2025, a team of researchers published a paper in Science Advances that, on its surface, had nothing to do with the Zulu kingdom or the Battle of Isandlwana. Its subject was a set of small stone tools, called backed microliths, that had been excavated from a sandstone overhang called Umhlatuzana rock shelter in the foothills of KwaZulu-Natal. The microliths were dated by stratigraphic and radiometric methods to roughly 60,000 years before present. They are among the oldest, deliberately hafted projectile tips ever recovered in southern Africa. The team did something that, until very recently, was not technically possible. They subjected the organic residues clinging to the microliths to mass spectrometry analysis, looking for the chemical signatures of plant or animal compounds that might have been applied to the stone. The detection thresholds for ancient organic residue analysis have improved by roughly 2 orders of magnitude in the last decade. What was invisible in 2010 is now sequenceable in trace quantities on a 60,000-year-old chip of stone. The signature came back unambiguously. The residues contained bufanedrine and a closely related compound called epibufanesine. Both are isoquinoline alkaloids. Both are produced by exactly one plant in the southern African flora, Buphone disticha, the Bushman poison bulb. The same plant whose alkaloids were prepared by the Izinyanga in Ketshwayo's war camp 5,300 decades later and given to the warriors marching toward the British column at Isandlwana. Stop and consider what this means. The pharmacological knowledge that traveled into Cetshwayo's army was not a Zulu invention. It was not a 19th century innovation. It was not even a Bantu innovation in the sense of having arrived with the southward expansion of Bantu-speaking peoples roughly 2,000 years ago. It is older than agriculture. It is older than pottery. It is older than every continuous cultural tradition currently known to anthropology. It predates the migration of modern humans out of Africa into Europe. It predates the last glacial maximum. By any reasonable comparison, the chemistry the British encountered at Isandlwana in 1879 was the longest continuous applied pharmacological tradition on planet Earth. The same plant, the same compound, the same valleys. The continuity is not metaphorical. The microliths were excavated from KwaZulu-Natal. The bulb still grows in KwaZulu-Natal. The descendants of the men who took it still live in KwaZulu-Natal. The chain has thinned in places, certainly. There were almost certainly long periods when the knowledge moved through small, discontinuous bands of foragers. There were certainly transmission events, lost recipes, regional variations, but the molecule the izinyanga handled in 1879 was the same molecule that had been deliberately applied to weapons in the same region 60,000 years earlier, by people who, biologically speaking, were not yet anatomically distinguishable from modern Homo sapiens by very much. The Umhlatuzana finding has done something specific to how ancient DNA and ethnobotany researchers think about Southern African pharmacology. Until 2025, the standard frame was that the ritual war medicine of the Zulu and other Bantu peoples represented a synthesis of relatively recent traditions, perhaps 2,000 to 3,000 years deep, drawing on plants the migrants encountered as they moved south. The Umhlatuzana microliths invert that frame. The plants and their preparation were already there. The migrants did not invent the pharmacopoeia. They inherited it. They walked into a region in which buphone disticha had been a known, applied, controlled hallucinogen and analgesic for tens of millennia, and they integrated that knowledge into their own ritual systems. The British thought they were fighting a primitive enemy. The Zulu warriors were primitive in no meaningful sense whatsoever. They were the inheritors of an unbroken pharmacological tradition that, in chronological terms, had been refining itself in the same hills for roughly the same span of time as the entire human occupation of Europe. The next question is the obvious one. What did all of that chemistry actually do to a man on a battlefield? The answer comes from the British and from their dead. The British camp at Isandlwana was struck shortly after noon on January 22, 1879. The first contact came at the northern flank, where a small mounted patrol pursued a few Zulu cattle herders to the lip of the Ngwebeni ravine and stopped, struck by what lay below them. 25,000 men, sitting cross-legged in disciplined silence, looking up. The Zulu commanders, Nchingwayo Kamahole and Mavumengwana Kundlela, had hoped to attack the following day. The patrol had forced their hand. The valley moved. The horns of the bull tactic, refined by Shaka in the 1810s, was simple and devastating in execution. The chest of the army moved directly toward the enemy. The two horns, made up of the youngest and fastest regiments, ran out wide on either flank, curving around behind the British position to close the encirclement before the chest made contact. A reserve, called the loins, sat with their backs to the battle to keep their morale unbroken until they were called forward. The whole maneuver depended on speed and on absolute disregard for personal exposure during the closing rush. The British firing line held at first. Martini Henry rifles, properly supplied, could put down sustained volleys at 400 to 600 yards. The advancing chest began to take heavy casualties. Period accounts describe Zulu warriors cut down in waves, the front rank falling, the men behind running over the bodies and continuing forward. This is the moment in the battle where the pharmacology becomes legible in the British record, even though the British did not have the framework to interpret what they were seeing. A man whose nervous system has been loaded with delta-9 tetrahydrocannabinol at battlefield concentrations, layered on bufenadrine, layered on doctored beer, will not respond to a bullet wound the way a sober man does. Bufenadrine is analgesic. THC at high doses dissociates the perception of pain from the body's response to it. Adrenaline does the rest. British survivors at Isandlwana and at later battles in the same campaign repeatedly described the same phenomenon. Warriors took rifle rounds and continued moving. Men with visible chest wounds reached the firing line. The wounded did not slow. The dying did not retreat. Officers wrote home about it. They called it fanaticism. They called it superstition. They called it the effect of dangerous belief in invincibility. What it actually was, in the most literal pharmacological sense, was a battlefield engineered through chemistry. The bodies of the Zulu warriors were fighting a battle. Their nervous systems were responding to a different one. The British line broke around 1:30 in the afternoon. The chest of the Zulu army made contact. The horns closed behind the camp. A solar eclipse darkened the sky over the valley as the fighting reached its peak. The British survivors were driven back into the camp, where they ran out of ammunition and tried to form into makeshift squares. Zulu warriors broke into the squares with Iklewa stabbing spears, the short, broad-bladed weapon Shaka had introduced 60 years earlier. The fighting at this stage was hand-to-hand, conducted with a weapon designed to be thrust upward into the abdomen at close range. There were no prisoners. The Zulu did not take them. Wounded British soldiers were finished where they lay. By late afternoon, roughly 1,300 men of the British force were dead. It was the worst defeat the Victorian army would suffer at the hands of an indigenous force in the entire 19th century. Lord Chelmsford, who had been chasing what he believed was the main Zulu army somewhere to the southeast, returned that evening to a camp full of corpses and broken supply wagons. One of his captains later recorded stumbling in the dark over the naked, gashed, and ghastly bodies of his late comrades. Chelmsford had not been outnumbered by men. He had been outnumbered by men whose nervous systems had been deliberately, chemically, ritually altered for the moment of contact. He never understood that. None of them did. For most of the 20th century, the genetic history of southern Africa was a tangled and largely unresolved problem. The Bantu expansion was understood in broad outline. Beginning roughly 3,000 years ago, populations originating in what is now the Cameroon-Nigeria borderlands moved south and east, eventually reaching the southern tip of the continent by around the 4th to 7th century AD. Beyond that, the resolution was poor. The Zulu, the Xhosa, the Swazi, the Sotho, the Pedi, the Tswana, the Tsonga, the Venda, the Ndebele were assumed to be one undifferentiated genetic component of the broader Bantu Southern wave. Whether they could be distinguished from one another at the level of the genome was an open question. In 2021, that changed. A team led by Driti Sengupta at the University of the Witwatersrand, working with collaborators at Uppsala University, Ghent University, the KwaZulu-Natal Museum, and the H3 Africa Consortium, published a paper in Nature Communications. The dataset was unusually large. Genome-wide data from over 5,000 living South Africans, drawn from 8 major southeastern Bantu-speaking groups, sequenced and analyzed at fine resolution. It was the first study of its scale in the region. The first finding was that the genetic substructure was real. The Zulu were not interchangeable with their neighbors. They clustered tightly with the other Nguni-speaking populations, the Xhosa, the Swazi, the Ndebele, and they separated cleanly from the Sotho-Tswana cluster and from the Tsonga, who fell on a separate axis. The genetic structure aligned, with high statistical confidence, with the linguistic phylogeny. The boundary between Nguni and Sotho-Tswana, which had been marked on language maps for over a century, turned out to be marked on the genome as well. The second finding was the one that made the paper a hinge moment. The researchers compared the modern data against 5 Iron Age farmer genomes that had been recovered from archaeological sites in northern KwaZulu-Natal, including one from a site near Champagne Castle in the Drakensberg foothills. The youngest of these Iron Age genomes was approximately 400 years old. The oldest was older. When the team performed the comparison, they found something that the existing literature had not predicted with this clarity. The genetic signature of the Iron Age farmers was statistically continuous with the modern Nguni population of the same region. Spell that out plainly. The men who lived in the homesteads scattered across what is now KwaZulu-Natal in the 17th century. The men who watched Shaka rise in the 1810s. The men who built the great kraals Lord Chelmsford burned in 1879. The men who drank Intelezi at Ulundi and walked toward the British rifles at Isandlwana were genetically continuous with the people who live in those same valleys today. The Y-chromosome lineages match. The mitochondrial signatures match. The autosomal signal, the bulk of the genome, match. The continuity is direct and measurable. The study went further. It identified differential Khoi-San admixture across the 8 Bantu-speaking groups, with the Nguni carrying somewhat less than the Sotho to Tswana, and it identified a sex-biased gene flow pattern that echoes the one reference script one described for Utsi's neighbors. The Khoi-San genetic contribution to modern Nguni populations came predominantly through women. The Y-chromosome line stayed Bantu. The mitochondrial line absorbed lineages from the older populations of the region. Men stayed, women came in from outside, the same pattern that has shaped small-scale agricultural societies from the Neolithic onward. The Sengupta paper did one more thing. It estimated the timing of major admixture events. Most of the Khoi-San gene flow into Nguni populations resolved to roughly 600 to 900 years before present, predating both the Mafeking and the Zulu kingdom by several centuries. The genetic structure that defines a Zulu man today was already in place when Shaka was born. Shaka organized it. He did not create it. The British buried the kingdom. They could not bury the genome. The next section explains, at the level of single chromosomes, exactly why. The Y chromosome is the only chromosome in the human genome that does not recombine. The other 22 pairs reshuffle every generation, mixing the maternal and paternal contributions into a new combination in the child. The Y is different. It passes from father to son virtually intact. accumulating only the slow, predictable mutations that arise from copying errors during the production of sperm. Every man alive today carries on his Y-chromosome, a written record of his direct paternal line, stretching back through his father and his father's father and his father's father's father, century after century into deep prehistory. The mutations on the Y can be read like a genealogical timestamp. They can be sorted into a tree. The tree has a name. It is called the Y-chromosome haplogroup phylogeny. The dominant Y-chromosome haplogroup among Zulu men and across the Nguni populations more broadly is called E1b1a. In some classification systems, it is written EV38. It is one of the most geographically informative lineages in the human genome. E1b1a originated in West Central Africa, roughly 25,000 years ago. Its sublineages began expanding rapidly between 5,000 and 3,000 years before present, in a pattern that maps almost exactly onto the archaeological evidence for the Bantu expansion. As the Bantu-speaking populations moved south and east out of the Cameroon-Nigeria region, they carried E1b1a with them. Today, sublineages of E1b1a dominate the male population of every Bantu-speaking region of sub-Saharan Africa, from Cameroon to Mozambique to KwaZulu-Natal. In modern Zulu populations, the frequency of E1b1a is high. Studies have placed it consistently above 80% of the male population, with several closely related sublineages dominating the local distribution. The mitochondrial picture, by contrast, is wildly diverse. Modern Zulu women carry an extraordinary range of haplogroups, including substantial frequencies of L0D and L0K. The matrilineal signatures of the Khoisan, the older population of southern Africa whose ancestors lived in the region for tens of thousands of years before the Bantu-speaking migrants arrived. The pattern is the same one referenced script 1 described in the alpine valleys around Otswurst, a tightly clustered paternal line, a scattered, diverse maternal line. The genetic signature of a society in which men stayed and women came in. Patrilocal, patrilineal, exogamous on the female side. What this means is specific. The warrior tradition the British encountered at Isandlwana was not just a cultural inheritance. It was a biological one, in a precise technical sense. The man who handed his son an Ikolowa spear at the end of the 18th century was almost certainly handing him the same Y-chromosome his own grandfather had carried. The man who handed it on in the 17th century was doing the same. The chain of fathers that runs through the Iron Age homesteads of northern KwaZulu-Natal, through the rise of Shaka, through the army that defeated Chelmsford, is a chain that can now be read directly from the cells of the men who live in those valleys today. A modern Zulu man living in Durban or Pietermaritzburg or Ulundi who walks into a clinic and gives a cheek swab for a commercial ancestry test will get back a result that includes a specific E1b1a sublineage. He may not know what the alphanumeric code on his report means. The code is the genealogical timestamp. It tells him that his direct paternal line traces, without interruption, through the same lineage that carried iron tools and Bantu agriculture out of West Central Africa thousands of years ago, through the Iron Age farmers at Champagne Castle, through the warriors at Isandlwana, into him. The line did not break in 1879. It did not break under colonial occupation. It did not break under apartheid. It is still there. It is still being passed. The pharmacology is older than the lineage. The lineage is older than the kingdom. The kingdom is older than the colonial border. The colonial border is gone. The lineage is not. There is a thing that happens with the Zulu warriors every few years, which is worth naming. A new technique emerges. A new sequencer, a new mass spectrometer, a new ethnobotanical survey. Researchers go back to the same valleys, the same plants, the same living people, and ask a question the previous generation could not. And every time, the answer makes the warriors more recognizable, not less. In 2021, the genome study. In 2025, the microlith chemistry. The pharmacology grows on the hillsides of KwaZulu-Natal. The Y-chromosomes ride buses in Pietermaritzburg. The mitochondrial DNA passes from grandmother to mother to daughter in households in Ulundi. The British buried the kingdom. They could not bury the people. 147 years after Isandlwana, the warriors are still walking. Please like and subscribe for more such videos. I will see you in the next one.